So Diana, do you need a lot of time? Do you have a long presentation? No, I'm just going to get started right now then, I think. All right, hi everybody. So let's go ahead and get started. Welcome to the first NBC Lunch Seminar of the academic year. For those of you who don't know, Michael Nachman is off frolicking. Sorry, he's off on sabbatical in Spain. And so during this year, I'm going to be standing in for him. So I'm the one who's going to be at the front of the room before these seminars. And because this is the first NBC Lunch Seminar, I just want to ask if people have actually enrolled in NBC Lunch Seminar. Those of you who've enrolled, anybody? Two? Well, that's enough. Encourage your friends to enroll in an NBC Lunch Seminar. Well, thanks to the two of you who have, and hopefully others who are maybe watching on Zoom have also enrolled in this. Because if we don't get enough, then it doesn't make it. We don't get credit for putting this show on. So the way this normally goes is we start by listing off the NBC seminars that are, sorry, the other seminars that are happening around campus. And I'm going to go ahead and do that unless anybody has any announcements they'd like to make beforehand. Nope. Okay. So the upcoming seminars for the coming week include the ESPM seminar, which is in 132 Mulford Hall. That one's tomorrow from 3:30 to 4:30. The speaker is Joshua Garcia, who's the president's postdoctoral fellow in the Department of Land, Air, and Water Resources at UC Davis. And his presentation is Building Sustainable Agro-Ecosystems from the Ground Up. We have an IB seminar tomorrow in 2040 Valley Life Science Building, and this one's from 12:30 to 1:30. And the speaker is Kyle Summers. Kyle Summers does work on poison frogs, and so definitely and you could be giving that seminar in NBC Lunch, right? So encourage people to go see Kyle give this presentation. The title of his presentation is Poison Frogs, Real and Imagined. It's very mysterious. Essig Brunch on Friday from 10 to 11. In 2063 VLSB is apparently meeting, but the speaker and the title is to be announced. And Botany Lunch in VLSB 1002 is also on Friday from 12 to 1. The speaker is going to be Brent Mishler, and he's going to be talking about reminiscences on 30 years as director of the UC JEPS and musings on the future. And then the NBC Lunch Seminar next week will be presented by Sean Harrington, who's a senior research scientist at the Wyoming INBRE Data Science Corps. And the title of his presentation is Population Genomic Responses of North American Snakes, the Past and Present Climate Variation. So normally I'd be introducing the speaker, but today we have a finishing talk, as you all know. And Diana's advisor, Rasmus Nielsen, is going to provide.
Yeah, thank you so much, Jim. So so as Jim mentioned, this wasn't advertised as a finishing talk, but that is what it is, even though Diana is not an NBC student, but she could have been an NBC student. She would fit right in here. The reason why she's not is she's in the Computational Biology Program, and there's not great tradition for computational biology students to actually become an NBC student, but we should change that. Because we are getting more and more students like Diana that do great, interesting biology, but come to that program. So I would say as a mentor, my greatest achievement I feel here at Berkeley is that I've managed to take some of these students that come in to the CCB program. They want to improve the human conditions. They want to work on cancer. They want to join medical research. But then I get them away from that and to concentrate on something much more important, to the evolution of frogs on this. I'm really proud of that. And Diana is one of those students that I've been able to. It took a few years, but a few years of brainwash, and now I have convinced herpetologists. So I feel like that's I'm very proud of that. Way of treating. So Diana, anyway, I'm talking about it's supposed to be about you. So Diana will be talking about one of her research projects, which is about the strawberry poison frog, and I'll let her talk about that. But I just want to mention that she's been doing a lot of other really cool research. She's very versatile and been working on very many different projects. So in one project that she's already published, she worked on the genetic basis of color variation in these toad-headed lizards from the Gobi Desert in China. Really cool study. And another study that's not published yet, she's been working on these Florida pumas, mountain lions, and looking at conservation genetics in them. They have this very famous story of that there was a lot of inbreeding, and then some humans from Texas were introduced there, and that led to genetic rescue. People arguing we now sequence the genomes, and Diana has some interesting results about what's really happening with the genomes of these water pandas. She has worked also on humans, where she has worked on projects of adaptation in some women from Korea to a lifestyle based on diving, when she worked on genetic adaptation there. And what else? Many other projects. You've been doing all kind of different techniques, in situ hybridization, very advanced computational techniques, all kind of different stuff. It's been a great pleasure to work with a student as intelligent and versatile as Diana. But I'll let you yourself go ahead and talk about the poison frogs.
Thank you, Vincent. Hi, thank you for coming. I'm going to be talking about my frog project. As Rasmus mentioned, I worked in many different things for my PhD. My thesis was like something very broad title, like adaptation in vertebrates, because I was working with very different things. But today it's all about the frogs, which is one of my favorite projects and the most colorful. So, okay, I'm having this problem again. Okay, so my system is a strawberry poison frog, Oophaga pumilio, which lives in Central America. This is its distribution. It goes from Nicaragua to Panama. And through most of the distribution, the coloration of this frog is like this, like red with blue limbs. They call it sometimes like a blue jeans morph, because it looks like it's wearing pants. And I think it's also like a little Spider-Man. But this is like a really cute morph, and it has a posematic coloration, which is like this warning signal because they are poisonous. So predators are supposed to recognize them and know they shouldn't be eating them. But there's like this very particular case for these frogs in this province in Panama that is called Bocas del Toro, where we have like many different islands, and in each of the islands we have like a different color, but they're all still the same species. But we have like a island that it's like green, another one that it's orange, another one here in the south that is also like a darker shade of green, and there's like some islands where we have more than one color. For example, this island that it's kind of like a shape of a triangle. In this corner of this island, we have a population that we call the cemetery hill. And there we have like polymorphic populations. We have like yellow and red frogs and many colors. But in the south of the island, it's like very isolated from this part of the island, and there's only red frogs in this region. And the other part where we have like polymorphic population is in this bay. This is called the Dolphin Bay. And frogs in this area go from blue to red. And I think for this talk, I'm gonna be like focusing mostly on those polymorphic populations, but I actually did analysis for a bunch of populations. Like my collaborators collected like 350 of these frogs. I did the library prep with Lydia. Thank you, Lydia. And then we did exome capture sequencing because the genome of these frogs is like 6 gigabases and it's super repetitive. So we decided to do exome capture. I think I actually talked about everything that I was going to talk in this slide from here. Oh, and another interesting thing about these frogs here is that, I mean, they're varied in color, but they also have like a lot of variation in their patterns. Like some of them are like solid color, some of them have speckles, some of them have like bigger spots. And also like the formation of this archipelago is super recent. It is like 1,000 to 9,000 years ago, and we see all of this diversification. So part of my project was trying to understand how all of these colors evolved and what's the history of these frogs and how are these like different colors produced. Oh, and this is just like when the splits of the islands occurred. And then I mentioned this, that we did the exome capture and we had like 350 samples. And I already talked about the sympatric populations, but here I highlight them again. So this I will be referring to as the cemetery population, and this one I will be referring to as the dolphin bay. So this is one of the first things that I did, that is try to understand what's the population structure in the island. So I looked like at the SNPs, like this map shows all the sampling locations for the samples that we have. And this is like a principal component analysis. I'm showing you the first 3 components. You can see that in the PC like actually reflects a lot of how the populations are distributed. For example, these here are these 2 of San Cristobal population. We have the cemetery population here. And you can see that like at least with these 3 first components, we cannot really separate the yellow from the red morph. And then here we have the hospital point, which is this one that is in front. And you can see it's kind of like here, this triangle that we have here. And this is called Tranquilo Bay, is the red frogs. And as I was telling you, like these frogs, they're actually on the same island, but they're like very far away. And then you can see that genetically, they're also kind of like differentiated, the ones from cemetery from the ones of Tranquilo Bay. And we also have frogs, for example, here in this island is called Popa. And most of the samples are from like the south part of the island, which are here. But like there's 3 samples that I've checked the labels, and they're actually the exact same samples that are here collected from the north part of the island. And you can see, like, for example, genetically, they look like they're closer to these Tranquilo Bay samples, which are like just across like this island. And I mean, there's like so many things that we can look in this PCA and against like the map, but it kind of not perfectly, but it kind of like reflects kind of like the geography of the populations. And then, yeah, again, I was going to highlight this. And then also like the Dolphin Bay populations, which are here that go from blue to red. And next to it, I also have like these shark hole samples that are kind of like in the same region, but they're populations that are only blue. And these ones are also called Rana Azul, like this area is kind of further, and they're also like just blue frogs, and you can see they're here. So they're very near to the Dolphin Bay ones, but they're kind of separate a bit. Okay, so then I did this population structure analysis using Ohana, where you define like different number of clusters and then see how like your different ancestral components go to each of your populations. I increased the number of K until like I was trying to see if I could get a component for each of my populations. I actually kept increasing from 9 and 10 and 11 to see if I could, like, for example, separate the Dolphin Bay populations or the cemetery hills. And you can see actually here with K9, the cemetery frogs start having like a different component, but it's not really separating red from yellow, but just like within the frogs. So it's kind of like showing that the red from the yellow are really not different from each other, like they're like the same population. And what else can I tell you about this? I made like these trees of these ancestral components. And one thing that I think it's interesting is that, for example, the green frogs, they're kind of like close together here in this tree that I made in relation of all of the different ancestral components. And I don't know if you remember, but geographically, actually, the green populations are kind of far away. Like some of them are here and these ones are here in the bottom. So I thought that was interesting. And then I was trying to like infer like the patterns of population splits and admixtures. And for this, I used 2 different programs. First, I used Trimix, which is like a very known software that a lot of people use. The problem here is that you have to specify the number of edges that your model has. Like you can start with like 0, 1, 2, like migration edges. And I have a lot of combinations, and I was trying to kind of like verify these patterns also with a 4 population test to see what makes sense. And I ended up with this tree that makes the most sense. And I mean, I think there are a few things here that I can point out. Like, for example, the migration edges that I found, I put here in this map. So like there's migration from Colon into Hospital Point, and from Hospital Point into the cemetery populations. And And then, for example, these two populations from here are like sister groups. And I mean, there's like many relationships that I could point out. And as an outgroup, I use imitator. But then I also decided to use another program that it actually, you don't specify beforehand how many migration edges you have. It just like converges like to what is the best number of migration edges. And it gives you like a posterior likelihood. So this is the topology that has like the highest posterior likelihood. And so this topology is very similar to, I mean, I know you don't remember just from seeing it, but trust me, it's actually very similar to the one that I showed you before. Something that changed a bit, and you will see, is that, for example, here, it's like migration from an ancestor of colon into the ancestor of these two. So kind of like this arrow that we have here, instead of just pointing here, it kind of points to the ancestor of these two. And then here, like this, we could like represent as having these two and migration from Tranquilo Bay, or having these two and having migration from this. So basically this arrow is flipped. So this is like the new one, but it's actually very similar. I know there's like too many colors and populations going on, so I'm sorry if this is a little bit hard to follow. But like this is like the relationship that I found and the migration patterns that I found for like these different populations. And then I was trying to infer also like the, yes. Yeah, some of it it does. For example, I mean, I think these ones in Ohana, they're actually not even separated as a different component. And these numbers are actually the number, like the support that there is. And you can see this is 100% of the time. Yeah, I mean, usually these three are kind of together. I think in Ohana, they're also together. I guess the difference, for example, is that Colon is kind of here in the middle with Popa, like the two green populations. So I guess that's different. Like here in this one, that is also similar. I mean, the thing is, like with the trees, you can flip them and they're kind of, so it's sometimes a little bit hard to see. But I would say like overall, yes, for example, this San Cristobal population is closer to this part, these ones in the bay here, so this island. It's kind of like there's like this cluster here, and it's like these populations. And then like the ones from here. And I think in Ohana results, it was like also like, see, this is like the San Cristobal ones, and these are like the ones from the Dolphin Bay. And these are the other ones that I was talking about, that they're kind of like in a triangle. So I mean, they're kind of similar, not exactly the same. Okay, so then I was using DADI to infer like how long ago it was a split, and I used like this very simple model where like it's just, I did it pairwise because it was like too much to try to do like all of the populations at the same time. So I'm just gonna show you an example of like one of the ones that I did. So I did like Colon versus the cemetery population. And the model was like, the split was 24,000 years ago. And you can see the split of the islands was like 5,000 years ago. And I basically did this for all of the combinations of populations. And in every case, it was like the split of the populations precedes the split of the islands. Which if we like talk about how like these islands were formed, it was like through like the last glaciation era. But we also know like there has been like several glaciations. So like we think it might be that they got separated before by a like earlier glaciation era than this last one that finally formed the islands the last time. Okay, so now I'm gonna talk about the genetics of the colors. And so for this, I was showing you, this is like the main morph we have, but in these islands, we have like, these are literally pictures of all of the frogs that, some of the frogs that I have for this study. And you can see like, we have like many shades of like red, and even like the red, some of them are like almost orange, and some of them start getting like really dark and almost like these are from the region that we have like blue and red frogs. So this is generated with DALI. It's blue jeans frog. I forgot I put that in there. And for this, I'm also gonna talk a little bit about like the cell types that produce different colors so that it's easier also to talk about this. So this is the thermal chromatophore unit, and this is the cell types that we have in the skin of frogs. And so it's the centrophores that are like the cell type of the type of chromatophore that produces carotenoids. So it's usually the cells that look like yellow or red or orange, and they filter the light. The iridophores that are like these reflecting pellets that scatter the light. So they're like little mirrors kind of, or I don't know how to explain it. And the melanophores, which are the ones that are producing melanin and are usually the ones that are producing like black or brown colors. Is like the ones that mammals have, and that's why we have only boring colors. But frogs have also centrophores and iridophores, which help them have like a bunch of different colors, and depending on how they're like laid out on top of each other, they can like sorry, I'm losing the words. It looks like it's a different color. So there's like this thing that, I mean, this is a visible light spectrum, and I only talked about like how we have centrophores and basically cells that look like mirrors and that are black, but how do we get all the colors? And the answer to that is like, it's a structural color, which means it's not like you're actually producing the pigment, but you're modifying how the wavelength goes through it, like usually through these two, iridophores and melanophores, and then that's how you get like these other types of wavelengths, and then the frog can look another color that it's not just yellow or black. Okay, so to see which colors, to understand like some of the colors, one of the easiest things was to try to look at these polymorphic populations, because they're basically not genetically differentiated between them. So from here, and we know like the ancestral color of these frogs is red. So from this population, we can like find out how do we get yellow frogs. And from here, we can understand how we get blue frogs. And here I did like pairwise FST, which is a measure of like allelic frequencies, and see how different the allelic frequencies are in one population versus the other. So if they have the same allelic frequencies, they would have like a 0 FST, meaning they're not differentiated. If they have like opposite allele frequencies, it would be 1, so they're super differentiated. So you can see here, so the FST between the cemetery population is basically 0, so they're not differentiated. And the same for Dolphin Bay. So we're gonna talk about now yellow frogs. And the fact that they're not differentiated like genome-wide is very helpful, because then I can do a genome scan looking for a region that is actually very differentiated between them, and hopefully find the gene that's causing it. So when I did that, I found like this pig here that has a bunch of genes, but the main one is this one that is called TTC39B. And also, like from previous studies from the lab that I collaborate, they did like these wild pedigree crosses that suggested it was like a single locus that is causing like these differences between the red and the yellow ones. And they said like red is dominant over yellow. So it was actually promising that we have like this hit. And what does this gene do? So like this paper is from 2022, and it's basically, they found that in birds, it converts like this dietary carotenoid. It enhances the conversion from yellow to red. You know, in birds, this same like group of genes is in the sex chromosome. So I think in some of them, and that's why in some birds, like some of the females, I think are one color and the males are the other color. But I don't know a lot about birds. But so the idea is that in these frogs, probably there's like some type of defect in this gene in the yellow frogs, that it's not doing that conversion, and that's why they stay yellow. And then like what's the genomic context of these genes? So we looked at the FST in this region, and these are like other genes that we find in the region, and you can see like it's not only that gene, but it's like other genes also have like kind of like high FST. One of the genes like, and for example, there's like this gene in the middle that doesn't even have high FST, but then we have this one that is STIRP1. And I'm gonna talk about that gene later too, but it's a gene that it's also known sometimes for patterning phenotypes, and it's like a gene that has been found like in coloration stuff. So I think it's interesting, and I'll show you later if I get to that part, because maybe I'm going too slow. And another gene is this PCIP1 that I'm just showing it to you because we also did a genomic association using red wavelength from images. So the scan that I showed you before, it was like using FST only with the cemetery populations. And here I actually use like all of the 300 frogs, like a measurement of how much red they have from pictures. And I did a genomic association. And then like the highest peak is like this one that has, I mean, it's kind of like the same region that has PCIP1 that I just showed to you. It's in the same region as TTC39B. And another gene that we found here is also KIT, that is also going to be relevant later for the story. So remember that gene. And then this is like an average genomic tree of all of the populations. So here, maybe it's not, it was hard to find like different colors for all of my populations because I have a lot. But the cemetery populations, I put the yellow ones in yellow and the red ones in golden. And I don't put them in red because I also have another red population. So sorry about that. But then if we look at the genes, for example, if we look at TTC39B and PCIP1, we can see like in the average genomic tree, they actually don't separate. But if you look just at the gene, the yellow ones cluster with the green ones, with the green frogs from Popa and Colon. And the ones that are cemetery red, they are closer to these like orange and red frogs. And it's kind of like similar for PCIP1, where the red cemetery frogs are kind of like within the red and the orange frogs, and the yellow frogs are with the green ones. Okay, so then I was trying to check if there was like maybe gene flow from colon, because also like to make green, and I'm also gonna talk about that later, but basically you need to overlay blue and yellow. So it would make sense that green frogs have the same version of this gene that is not producing red, but it's like more like yellow. So I did this test that is like an ABABA test genome-wide, and like the p-value was significant to see if there was more patterns from colon with the red ones or the yellow ones. And then we got more patterns with the yellow ones, which suggests maybe there was like some gene flow there. So like this one is actually also like the model that I showed you before from the populations from tree-mix and admixed surveys, that there's like migration from colon into the ancestor here. So maybe like the allele like comes from there. And then I also did an HKA test. And we found that there's reduced polymorphism in both colon and the cemetery yellow ones, but not in the red morphs. So this is kind of like a signature of selection going on in the yellow one and the green one. So now I'm gonna talk about the blue frogs. So for the blue frogs, okay, I'm gonna use the ones in this peninsula. And then I did the same where I kind of like did this scan where I used the two frogs from Dolphin Bay and then an outgroup, and then look at the branch that has the blue frogs. And then the highest gene here is KIT. And then I also did like a genomic association using all of the frogs. And you can see here also like the highest gene is KIT. So what does KIT do? So KIT is a gene that is involved in melanogenesis. It's here. So we're talking about blue. So why is there a gene that is producing melanosomes, which I told you was like for black? So actually my collaborator did this for her PhD. She measured the different types of cell types between the different colors, and she actually found that the blue frogs have a lot more melanosomes. So it actually makes sense that I find this gene. And then before I was talking about structural coloration as well. So so this is just like a drawing on how like basically this is like the skin. And then the way that like the different like layout, probably having more melanosomes, is filtering these frogs, is making them darker and look blue, even if they're not really producing blue pigment. And then here I also did like some trees. Here you can see the dolphin bay. It's also like kind of hard. It really doesn't separate the ones that are blue from red. And it's a bit hard to see in this tree, but basically all of the blue frogs are here. And this is the same tree, but I tried to kind of like put in gray all of the other populations that we don't care about for the moment and just highlight the blue ones and the red ones to show that it's kind of like all of the blue ones have like this different version of KIT. And then I tried to look for synonymous and non-synonymous mutations and actually found 2 non-synonymous mutations in KIT, and this is how they're segregating in the different populations. Here I have like this pie chart show like the allele frequencies, and for example here the last letter of this code is the color, so these are red, intermediate, these are blue frogs. So you can see like the blue frogs kind of have like these other alleles in these mutations that are non-synonymous, and this is the KIT protein, and this is where they are. So I think this is transmembrane, and I think this one is like the one that sometimes binds like within the cell to some things, because KIT is a transmembrane protein. And then I did a tree also of like the different versions of the KIT, and then here like where are like each of the mutations, and you can see like all of the brighter colors kind of like don't have any of these mutations, and kind of like my theory is like with one of the mutations you become a bit darker, and with the other mutations then you become blue or something like that. This is also just showing more of the frogs and kind of like trying to show like the red that I have in some populations is a much brighter red. And then the red that we have in like the Dolphin Bay area is kind of like much darker. And many of these frogs actually already, even if they're red on their dorsal coloration, they're already like blue in their ventral. And I think maybe these 2 mutations are like playing a role there. We also looked at like the pairwise differences and Tajima's D, and I also did an HKA test, and all of these show that there's kind of reduced polymorphism in the blue more than in the red morph. This is only using like Dolphin Bay populations. But when I did the HKA test, I actually it was not significant in the Dolphin Bay ones that KIT has reduced polymorphism, although if you look at the numbers, it clearly has less. But when I did the test and corrected genome-wide, the Q value is not significant. But if we look at the shark hole, which are also blue frogs that are kind of like next to the Dolphin Bay, the test was significant there. So now I'm gonna talk about green frogs. What time is it? Okay. So I also have pattern, and I probably should go faster. So how are frogs green? So I mean, there's like, well, I didn't include that slide for a reason, so I'm not gonna talk about that. I'm just gonna talk about the frogs that are producing it, not through, there's like some frogs that produce it through a pigment, but this is not one of those frogs. This is the one that produces green through structural coloration. So this paper in 68 kind of shows an example in the tree frog where we can see centrophores, which are like the yellow cells, the iridophores, which are like these little mirror things, and the melanophores, like the black cells, and how kind of like with iridophores and melanophores, you produce like this structural blue coloration. And then when you overlay these 2 colors, you end up with green. And that's like what basically this paper says from 68. And I think also the last time I was here for a herp group, I even brought like these jars you have here in the MVC, because some of the frogs, when they wash out, like these really bright green frogs, when they wash out kind of like the centrophores of the colors in the jars, they end up being like basically this blue. So you can like see there like a demonstration by eye that actually produces a blue that it looks like this. I didn't bring a jar today, sorry. And then, but here it was a bit more challenging because I didn't have any population that was polymorphic where I had like green and at the same time I had red or like another color that was like basically not genetically differentiated. So I couldn't use like the same technique I used before. So what I did was basically compare the green frogs against red population, and then a green frogs against the blue population, and then look at the green branch, and then look what of those two had in common. So basically this, and then I was trying to see which genes are here, like having kind of like these different allele frequencies from these two, and then they're also common in the two. And then what I found is TTC39B again, which, as I said, I think it makes sense because I think to produce green, you need yellow. And the other gene that I found is like BCO1, which is beta carotenoid 1 gene. So what does this gene do? It cuts carotenoids into smaller carotenoids, or the different types of carotenoids. I forgot the names, but one of them I think is apocarotenoids, and then another and then depending on which types you have like different colors. And this is also something that my collaborator did for her PhD thesis. She measured the different types of carotenoids in these frogs, and she found that actually like the different color morphs have different content of carotenoids. But the thing is, like, carotenoids we usually just associate to like these red, yellow colors, right? And I'm trying to look at green. But then I was looking at like some papers, and then I found that in general, like more carotenoids means green in frogs. So these are like two papers in different species of frogs where they change the diet of the frogs. If they are like poor in carotenoids, they're like much pale. And then as they increase the number of carotenoids in the diet, they get like a lot brighter green. So I think carotenoids have to do with green as well. And then my collaborator did, she measured the amount of carotenoids, and you can see green frogs actually have the same, if not more, carotenoids than red frogs. And I also looked at the differential expression of this gene in like this publicly available data set. This is actually not, it's like from the same populations of Bocas del Toro, but I didn't generate this data set. It's like another group. But I looked at this gene, and BCO is actually much highly expressed in the green frogs. And then this was a bit confusing because I tried to look at non-synonymous mutations in this gene, and I actually found 4 non-synonymous mutations in this gene. And here are like the green populations. So I think we can see like there's like a pattern, for example, like this mutation maybe, like, or these two maybe are doing something to make them green, but like this mutation is also segregating, like it's also almost fixing this blue population and in this red one, so I'm a bit confused. But I found a bunch of non-synonymous mutations in this gene. Okay, and so how are all these colors evolved and maintained? So I already showed, like I did some tests in some of the populations, and we see signatures of selection. And then there's like these frogs and these populations has been studied like a lot, because a lot of people are kind of like impressed like how many colors you have and in such a short amount of time and stuff. So like genomically, I don't think anybody had done this, but they had studied them, like their behavior and like stuff like that a lot. And there's like papers where they do like female choice, and they've seen, for example, that in polymorphic areas, females usually prefer the males that are their own color. So for example, if you have yellow and red frogs, the yellow females will prefer the yellow male. And that kind of like could be driving a bit like the selection through sexual selection. And not only, I mean, there's like a lot of things going on, because it's also like a posematic coloration. So in theory, predators should also be playing a role. So it gets super complicated. And there's also male-male aggression. And with male-male aggression, it also happens that males actually only recognize males their own color as a threat. So they're only like aggressive towards men their own color, males. So it's kind of like very complex, but there's selection going on there. And then, do I have time to talk about pattern? What time is it? Okay, maybe we'll do this quick. So then I also wanted to know how the different patterns were formed, and like for this, this was not the original aim of the project, but like my collaborators sent me like all of these pictures of frogs, but they were not standardized at all. They were like not taken with like the same light source. So basically they were really hard to work with. And me and an undergrad, we just scrubbed all of these pictures of all of the frogs that I got sent to. And I was like, I'm gonna try to extract the pattern out of this. And the way that I did that is using this kind of like pixel thresholding method. So we got the distribution of pixels, and then from points, you say like all of these pixels I turn to white, all of these pixels I turn to black. And then I also ended up excluding, it has two lines, my distributions, because some of it excluding the glare. I was trying to, because the pictures were really not meant for this. So we tried to kind of like fix that and like kind of exclude the regions that don't have pattern. And like, this is kind of like what I managed to extract from these pictures. It's not perfect, but I mean, it kind of works. And then from this, I also created like this phenotype that I call black proportion, that I was kind of like saying how many black pixels out of the total pixels I had. And then I had like this phenotype that I could use for associations, for example. So one of the first, and I had to exclude all the dolphin bay populations because they were way too dark to make this thing work, and the pictures were not good for it. So I only used these populations and excluded everything from dolphin bay. And one of the first things we can see is that the pattern is really not the same in the different populations. And then, I mean, even if you look at pictures of a lot of frogs, for example, these ones are from the cemetery, they have like these more like coarse patterns with bigger dots. These are from Popa, they have usually little speckles, and other populations, they're usually like, I mean, there's variation within population, but there's also like more, like these patterns that are like bigger dots, they're usually from Colón and the cemetery and the yellow frogs. And this is relevant because remember I was showing you this gene before that is called TIRP1, that is actually in the same locus as TTC39B. So I thought that was interesting. And also, like, even if you compare yellow versus red, it seems that maybe the yellow frogs have more patterning than the red ones. So then I also did, I used a convolutional neural network. It's pre-trained, like you can like kind of load it in Python. It's like this CNN that has been trained with like millions and millions of pictures, not necessarily of frogs, like just of everything. And it's really good at recognizing patterns. So I inputted like it's already like these versions of my pictures that are only like binary, like with this like black or white thing. And I try to make it extract the principal features with this network. And then I do a PCA to like kind of like plot the features that, I mean, the kind of the variables that explain the most variance of this feature. And I end up with these two PCs. And you can see that it actually does like kind of like a decent job separating these frogs by pattern. And I did the classification using these binary images, but then I added back the color and I realized it's really not, like, it's also kind of like picking up the color, even though like, and the reason for that is basically because pattern and color in these frogs are not really independent from each other. And I also kind of like show that because it depends from which population you are, they're also different, having different amount of patterning. So what I did is like I did a bunch of genomic associations. I did it with the black proportion phenotype, and I also did it with PC1 and PC2. I'm sorry, like this is all very small. But here the main peak that I have is TTC39B peak, which is the same one where there's TIRP1. And here you probably cannot read it, but there's also TIRP1 in this association that I did. So I started thinking like maybe this gene is actually important for pattern. And then I also did like this correlation matrix between like the different wavelengths that I have, the black proportion PC1 and PC2. And then you can see a lot of these things are correlated with each other, the phenotypes. So like, it's just like more of the same, that the color and pattern are not independent. And then particularly for TRP1 and TTC39B, they're in the same locus. And then I found like in fish, there's these papers, for example, in this one, they talk about a TRP duplication and how this is kind of like responsible for the black patches. And that it's always like comes with this like orange phenotype, like, but they don't know where it's coming from. They don't talk about TTC39B in this paper, but my guess is like it has a role there. And then in this other paper, also in fish, they kind of like found that TRP1 is causing these black spots and TTC39B is causing the pink, but they actually don't ever mention that these genes are in the same region. I mean, I think for the reference genomes of many like organisms, like you don't have them in the same region because they're so fragmented. I actually just realized, because for my frog, they're not in the same genome, but then when I was looking information about these genes, I found them there together in the mouse. And then I looked them and they're also together in Xenopus. And then I realized they're probably together everywhere. So I think maybe these two genes are doing something there with the pattern and color. And then, well, I think maybe I don't have time for this, but basically I found a gene that's maybe causing these dots that in humans cause also like these little speckles, and but I don't have a lot more like evidence or analysis for this. It was just like a hit that I actually got for these frogs and for like the green wavelength. But I think like because all of these frogs are always having these speckles, it could be that it has to do with the pattern, because this is like a syndrome that is caused in human by that gene. And this is one of like the phenotypes that people get, like these little speckles in their mouth or in their hands or in their feet. So I'm done. So the takeaway is that the split of the populations precedes the split of the islands. KIT seems to be responsible or related in structural blue in these frogs. BCO is differentiated and more expressed in green frogs. TTC39B locus has a role in red versus yellow frogs and also in green frogs. Color and pattern are not independent. TRP1 and STK11 are candidates for pattern formation. And there's positive selection acting in some of these genes. And I think that's it. So thank you, well, all of you for listening to my talk, and also to my lab and all of my collaborators and the other undergrads that have been working with me, to my funding. And I mean, now I'm a postdoc in UCLA, so I changed my email here. And yeah, that's it. I mean, I had some slides that I pasted. I did kind of like this exit talk a couple of months ago in May for my program, but it was like a 15-minute talk, super fast. And I used like half of it for acknowledgements, but I left these pictures in the end. So just to thank my family, my friends in Mexico, more friends in Mexico, my friend Jessica and her emotional support dog, friends from the lab and from the Bay, friends from CCB, my program, and Carmel, which is like a really good friend of mine. And thank you. Do you have questions?